I thought I'd do a video about DNA and RNA nucleotide structure and mnemonics. So here are the structures of the bases, or the nucleotides rather, for DNA. The nucleotides are deoxyguanylate, deoxyadenylate, deoxycytidylate, and deoxythymidylate. So there's different names for different parts of these structures. The nucleoside, what it's called, is this part in pink here. So that's without the phosphate. A nucleotide is when it has one phosphate or more on this 5' position. So the way it's numbered, 1', 2', 3', 4', 5', the carbons are numbered. The reason they have prime in the name is because if you'd say without the prime, it's referring to the positions in the ring of the base up here. So this is the base, this is the sugar. A nucleoside is made up of a base and a sugar. A nucleotide is made up of a base, a sugar, and a phosphate. In DNA, the sugar is deoxyribose. In RNA, we'll see that it's ribose, so in DNA, it's missing an oxygen here on the 2' position. Professional biologists even will sometimes use the word nucleoside or nucleotide or base interchangeably, which can get confusing, and if you hear that in a professional biologist's talk, hear them use it wrong, you can laugh at them and think that you're smarter than they are. Depending on the context, people mix these up, and it's actually not a big deal. Here are the nucleotides for RNA, and you'll see the difference here in the bases is there is a uracil instead of the thymine. So the only difference here between thymine and uracil is this methyl group here. So the things that do the base pairing, the nitrogen and the oxygen that do the base pairing, are exactly the same. The only difference is a methyl group. The difference in the sugar, ribose instead of deoxyribose, is that there's this oxygen here. And that means that the RNA is actually more reactive. A hydroxyl group can react with things much more easily than just a saturated carbon. And you'll notice here some of these bases are called purines. These ones with the double ring structures, that's called a purine, and then the ones with the single ring structure, that's called a pyrimidine. So the way that I remember the DNA bases is with this mnemonic GACT. So I order them G-A-C-T. Lots of different people order them in different ways. Just whatever is easiest for you to remember. But I like GACT because I can pronounce it. And the best way I found to memorize anything is just to associate it with something goofy. GACT sounds kind of goofy, and so it's something I can remember. Another convenient thing about saying GACT is that these first two are the purines, and these second two are the pyrimidines. And the first purine in the sequence, G, base pairs with the first pyrimidine, just like this. So the G will base pair to the C, and the A will base pair to the T. And this just shows that there's 3 hydrogen bonds between the G and the C, and there's 2 between the A and the T. And then if you want to remember that the purines come first, if you need some way to remember, this gets kind of complicated. Maybe you won't even be able to remember this. But the way I remember it is that the U comes before the Y in the alphabet. So GA comes before CT in the sequence, and purine comes before pyrimidine in the alphabet. To remember the RNA bases, I just change that same, keep the same order, and then change the T to the U, and so I get GACU, which is also goofy and easy to remember. And the same thing goes for the base pairing. The U base pairs to the A, and again the purines are in front and the pyrimidines are afterwards. So to remember that the single ring structure is a pyrimidine and the double ring structure is a purine, what I do is I notice that pyrimidine looks like the base of a pyramid. Looks like the base of a convex hexagonal pyramid, to be exact. This is not really looking like what we think of when we think of a pyramid, even though it is technically a pyramid. It's a concave pyramid. So because GC bonds have 3 hydrogen bonds, they're actually stronger than AT or AU bonds. So here we can see that thymine and uracil actually have an extra oxygen here that doesn't participate in hydrogen bonding. But hydrogen bonding occurs when there's a partial negative charge on these oxygens and then a partial positive charge on these hydrogens attached to the nitrogens, because the nitrogen is pulling the electrons away from the hydrogen. And you'll notice that this methyl group does not participate in the hydrogen bonding, which is why when you remove it and you get uracil, it hydrogen bonds the exact same way. Okay, that's all. Thanks, hope it was helpful.